Well, those were some great talks, hard acts to follow, but I hope today I will be able to convince you that childhood leukemia research and treatment has really been a success story over the past 50 odd years and has really been at the forefront of not just childhood blood cancer research, but cancer research and treatment in general. And many techniques that we use now, such as molecular genetics, cytogenetics, minimal residual disease monitoring, started with pediatric leukemia trials. So as we celebrate 50 years of this department, and when I was thinking about what to talk about, I decided I won't really spend much time talking about what we do in our lab, but celebrate the last 50 years of childhood leukemia research and treatment worldwide, and I'll tell you a little bit about what's been going on in the UK. So let's think about what childhood leukemia or a child with blood cancer went through 50 years ago. So in the early 1970s, so 50 years ago, if out of 100 children who developed leukemia, at the end of 6 years, there would be maybe 30 who survived. And if we fast forward, I just want to show you that there has been incredible progress in the survival rates of children with blood cancers. And by the 2000s, over 90% of the children with leukemia could be cured. What's even more remarkable is that if we go back a few years before that, 1950, 100% of children with leukemia died within an average of 3 months of diagnosis. So when you look at these graphs, I think everybody will agree that this really is a remarkable success story. And I want to tell you the story of how we went from 0% to close to 100% in a little over 50 years, and what we can do to push to reach that last gap. But what is childhood leukemia? So as many of you know, all blood cells are formed in a healthy bone marrow, and that gives rise to the different kinds of blood cells we need: red cells, platelets, and white blood cells. As hematologists, we're used to looking down a microscope and looking at normal blood and bone marrow films. And this is what a normal bone marrow looks to us under a microscope. So this is a happy blood cell factory where you have many different kinds of cells that will eventually give rise to all the blood cells we need. This is a heart sink moment for us as pediatric hematologists when we look down a microscope and we find the different kinds of blood cells have been completely taken over by only one kind of blood cell, which are the leukemic cells. And leukemia originates from the Greek words white and blood, so white blood. When this happens, the normal bone marrow cells are obviously taken over and disappear. And what happens to the children is that they develop anemia, bleeding, and infections as a result of that. And in the 1950s, this is what the children were dying of, particularly bleeding, because platelet transfusions had not been developed then. So I want to briefly go through the timeline of childhood leukemia research and treatments. So Sidney Farber was the first person in Boston who actually tried aminopterin for these children who had acute leukemia. This is a folate antagonist, and interestingly, he actually first tried to cure them by giving folic acid because the bone marrow looked very much like pernicious anemia where there were loads of immature cells there. And he found the children actually got worse. So he tried a folate antagonist, and that seemed to work. Very shortly after, 6-mercaptopurine was discovered, and also corticosteroids and another drug which also prevented RNA and DNA synthesis, methotrexate. So these drugs were given singly to these children, and they evoked a transient response, but none of the children survived. So the clinicians then started trying combinations of these drugs, so combining methotrexate with 6-mercaptopurine. And this did lead to some success, but the children still died within 1 year of diagnosis. And sometimes it was because the leukemia came back in the blood, but a lot of the times the leukemia came back in the brain or the central nervous system. And this proved to be a big problem for clinicians dealing with childhood leukemia. The transformative thinking really came from a clinician called Donald Pinkel at St. Jude's Children's Center. And I believe St. Jude is a patron saint for hopeless causes, so it was an apt place that this was discovered, who advocated total therapy. So the aim was no longer to palliate or give these children a little bit extra few months, but to actually eradicate the leukemia so it didn't come back. So he advocated central nervous system radiation, so irradiate to prevent the leukemia coming back in the brain, and give several blocks of chemotherapy to eradicate the leukemia. And in early 1970, actually, the group reported that out of a small cohort of 39 children, 50% were cured. It was met with outrage. People told them that you're giving false hope to parents. It is a hopeless cause. But thankfully, they persisted. And around the same time, many other countries, such as the UK and Germany, started systematic trials of combination chemotherapy. So the 70s were actually an exciting time for childhood leukemia treatment. It was around that time that clinicians realized that CNS irradiation was not a good idea in children with growing brains, growing nervous systems and cognitive functions, and that it led to a lot of toxicity. So it was decided that CNS irradiation could be substituted by intrathecal chemotherapy and also more intensive blocks of chemotherapy, so we could do away with cranial irradiation. It was also a time where better supportive care was available, so blood and platelet transfusions were available, infections were treated better, and children started to do much better. ALL was also, acute lymphoblastic leukemia was also subclassified, so you knew they could arise from a T cell or a B cell lineage. And cytogenetics, or the study of chromosomes and how they could be abnormal in leukemia, was discovered around the same time. And in my mind, that really is the start of personalized medicine in childhood leukemia, because we quickly understood that not all leukemia is the same, and chromosomal abnormalities conferred a particular risk type to some leukemias. But by the mid to late 80s, 70% of children were now being cured with a combination chemotherapy and some amount of risk stratification. The 90s brought monitoring of residual disease, so you could actually sensitively monitor if the disease is clearing fast enough and well enough. And also treatments were refined. So by in the 2000s, remarkably around 90, we were hitting almost 90% cure rates in children with leukemia. Around the 2000s also, it was possible to study extensively the genetic and gene expression changes in different types of leukemia, which allowed us to actually develop targeted therapies. So what about the UK? So the MRC Working Party on Childhood Leukemias was established in the 1970s, and there were systematic trials, initially called UCAL 1 to 11, and then ALL 97, 2003, and the most recent trial that has just ended is UCAL 2011. And each of these trials led to increments in childhood ALL survival. An AML working party was also established, and although AML survival rates have been lagging, there also has been incredible progress from 6% up to now 72% survival. So the graph on the right shows survival rates for children with all kinds of leukemias, AML and ALL. And you can see from the 1970s, so in the last 50 years, the survival rates have almost tripled from 30% to 90%, which really is remarkable. So what do we do next? Where do we go from here? I think one thing I want to highlight is the age group in which childhood leukemias develop. So these are people at the very early stages of their lives. And especially for ALL, there is this huge peak in the 2 to 6-year-olds. So what we must remember that cure is not enough. They have their whole lifetimes ahead of them. So we need to cure them so that they can live a healthy life which has a good quality with not significant morbidity and toxicity. So rightfully, the clinicians and scientists have moved towards treatment reduction, more personalized treatments, more targeted therapy, and international collaborations. The current ALL and AML trials in the UK are both international collaborative trials. I want to just focus on the last 20 years, so from 2000 onwards, what progress we've made. So stem cell transplantation came to be in use, and it is a technique where you can get rid of all the diseased marrow and transplant somebody else's stem cells, which can take over and function normally. It's not without its own problems and toxicities. In the 2000s also, we started trials where treatment reduction was implemented. So the children who do the best, the standard risk leukemias, treatment started to be reduced. Immunotherapy appeared on the scene around the same time. And there can be different types of immunotherapy, but the basic idea is you identify a target on the cancer cell, which can be then targeted by different mechanisms. It could be an antibody that signals your body to kill the cell. It could be a bispecific antibody that recruits an immune cell like a T cell, which will then destroy the cancer cell. Or it could be CAR T cells, which many of you might have heard about, in which the patient's own T cells, and more rarely somebody else's T cells, are engineered to recognize a specific protein on the cancer cell and destroy that cell. So CAR T cells actually had been in the making from late 80s, but initially for HIV and solid cancers unsuccessfully. It was only in 2011 that it was shown to be effective in B-cell leukemias and lymphomas. And by 2019, it had been licensed in the US, the EU, and other countries. So this, including the UK. What other treatment options developed? So there are drugs now that we can use to target specific pathways in a cancer cell, so much more targeted and less toxic. And this really has been an exciting time the last 10 years or so. And I feel really privileged to be part of this journey. What we need to do now is wait and see the long-term consequences of these new treatments. I want to next talk about a few of the greats in the UK who really, the giants whose shoulders we stand on. So Prof. Hardesty was, I would say, probably the first pediatric hematologist, designated pediatric hematologist. And he really was instrumental in understanding prognostic factors in ALL, how central nervous system disease arises, why chromosomes are important in leukemia and how they can be abnormal. And along with Prof. Secker Walker and Prof. Christine Harrison, who's still instrumental in all the ALL trials, cytogenetics for risk stratification. Prof. Judith Chessels was really at the forefront of the MRC working groups and drove the clinical trials in the UK. Prof. Mel Greaves, who was the first person who actually proved that childhood leukemia occurs before birth. And even though that first hit happens before birth, it doesn't develop into leukemia till there's a second hit in childhood, which could be an infection. Prof. Irene Roberts, who is in this room, has really transformed how we understand prenatal and neonatal hematology and the pathology of Down syndrome AML. Dr. Nick Golden was instrumental in minimal residual disease monitoring, and this has really transformed how we treat children with ALL now. We're able to use very sensitive techniques to know whether a child is responding to treatment and adapt the treatment based on that. Prof. Brenda Gibson leads the childhood AML trials in the UK, and Prof. Moorman has been at the forefront of risk stratification of all children in the UK with ALL. Prof. Bhaskar Saha has been the lead for many of the relapsed ALL protocols in the UK. So we've reached where we are, and I hope you agree that it has been a success story because of the work of many, many people, clinicians, patients, scientists, donors, funders. And in the UK, we have been very lucky with, to get backing from some fantastic funding bodies. I have listed a few here, but there are many, many more who fund research and fund resources like the Childhood Leukemia Cell Bank, which we have used as researchers and clinicians to understand leukemia better. So where do we go from here? Can we reach the 100% cure rate? Perhaps we can, and that's what we are aiming for. But we also need to reduce toxicity. Perhaps in the future, we'll see chemotherapy-free regimens. Could we move to early detection and screening? This raises its own problems of who to screen and what do we do after screening. Perhaps we could, there are newer techniques such as gene editing and immune modulation that could be used. And the ultimate goal, and we don't know if this is possible, but some do believe that maybe one day we'll be able to prevent childhood leukemia. And lastly, I want to say we need a global strategy because 80% of the childhood cancer burden is in low and middle income countries where the survival rates are far, far behind the high income countries. So a lot of people ask me, childhood leukemia is a success story. Are you not done? Are we done? I like to think of it in my reflective moments of my own life and what I have learned during my life as a child, as a doctor, as a pediatrician, and then a scientist. So this is me growing up in the 70s and 80s. Around that time, childhood leukemia survival rates was 50%, as I told you. This is my son growing up in the late 2000s. And when I reflect, it's remarkable that the survival rate is almost double within that one generation. But when I mentioned, these are the survival rates of a child growing up in the UK. I grew up in India. And the reality is, when I was growing up in India, the survival rates for childhood ALL was probably less than 10%. And even when I graduated as a doctor, and I was seeing children with leukemia in a tertiary teaching hospital, we could only save about 30 to 40%. And the big problem was we didn't know the true incidence of the disease because there are children who are undiagnosed, children who are not reported, there's no proper cancer registry. And once you diagnose, 1/3 opt for no treatment because they cannot afford it. And many drop out once they've started treatment. So a hugely frustrating scenario. So I never failed to reflect that it was just by fate that my son was growing up in a country where rates were around 90%. And at the same time, probably survival rates were half in India. This has improved over the years, and a lot of it by efforts through some excellent clinician scientists such as Prof. Saha, who has moved to India now and set up really standardized, risk-adapted protocols that he's trying to roll out nationally. But there still is a gap, and we must address this gap. So when people ask me, are we done? I think the answer is we are not done till everybody is done. And it is achievable. I want to just end by saying where we fit in this picture. So just on the last slide about what we do. So we, as a group, try to understand how childhood leukemia develops before birth. What are the properties of the cells that transform? Which cells transform? Are they different at different ages? And use the answers from these to develop leukemia models that will help us develop novel therapies, especially for that 10% we cannot achieve cure for. And the 50% of babies, which is not a very well-known fact, but babies with leukemia do not do well. So our focus is very much on the high-risk leukemias where we are still trying to achieve cure so that eventually we can reach that 100% target. We want to do this as a clinician myself and through some brilliant clinical colleagues through the clinical trials. We have set up a partnership with India focusing mainly on infant leukemias and have many other collaborators throughout the world. At this point, I want to thank the many, many funders who have funded basic research in our lab, translational and clinical research, and also international collaborations. This is us, currently the team, and I'm very grateful to be part of this inspiring lot of scientists, some of who are also clinicians. And we believe that together we can make a difference in childhood leukemia. And I'm very, very excited to see what the next 50 years bring. Thank you.
Thank you very much for this overview and of a pulse taking of progress in pediatrics over many years. And clearly, we're not done yet. We all invite you in 50 years to come back and see what the progress was in the next 50 years.